What's up, everyone? Today we're talking about membrane transport. By now, we've made it clear that the cell membrane is more than just a barrier. It serves a lot of functions in the cell, including signaling, transport, movement, immune protection, cell-cell communication, and so on and so forth. We've also looked at how it is a fluid mosaic of proteins, lipids, sugars, and a lot of other items. So it is a complex structure, but after all, it is a border. It is a border between the inside and outside of the cell. What does that mean? It means that it needs to protect some things from entering the cell, right? And it needs to let some things through. So that's why we call it a semi-permeable membrane, right? It lets some things through, but not all of them. But it's not that easy. It's not that simple, right? In order to let through things at the right time, we have to have a few um, mechanisms and a few ways, right? It's just like letting things through. We're gonna have letting different things with different sizes through the doors or into the house. We're gonna have different types of doors depending on the shapes and the sizes and the qualities of the things that we want to get through. So these are different types of membrane transport. So let's look at a few of them in this video. All right, so this is a nice overview of all of them, and I like this uh, figure. So we're going to start with the first one, which is the simplest one, which is just simple diffusion, right? If you have small non-polar compounds, they can just go through. Why? Well, because the membrane, especially the inside of the membrane, is non-polar. So if you have small non-polar compounds, they can just go through, right? So this is called passive diffusion. What drives it? The concentration gradient or the concentration difference between the two sides, right? If you have a low concentration here and a high concentration here, it's going to move, right? It's going to move towards the low concentration. So this is the passive diffusion, right? And passive diffusion moves differently, right? We can have it driven by concentration, but we can also have a concentration gradient, but also we can also have it driven by charge gradient, right? So it can be either a concentration or a charge gradient. An example is oxygen. Oxygen and CO2, they go in and out of the cell by using simple diffusion. These are small non-polar molecules that can do that. Right? So this is how it goes into the cell, like in the lungs and in the red blood cell. This is how they go in and out, just by simple passive diffusion, right? So for each one of these, I'm going to explain it, show you how it works, and then give you an example. So you should be able to do that for each one of these as well. All right, let's go to the next one. The next one is facilitated diffusion. So it's still diffusion, but it is facilitated, right? So we're using a gradient as well, right? So there's a gradient. If there is a gradient, a concentration gradient, then things are going to move down the electrochemical gradient. So electrochemical, that's why I was talking about. It might be electricity or charges or just concentration. This is the gradient we're talking about. But this is facilitated. It has a transporter. An example is, let me just give you this nice video here to show you what's happening, right? This is just a facilitated diffusion. It uses a channel, right? So this is just a channel. Notice that this channel is not moving at all. It's just there. And these are moving in and out. So aquaporin is an example, right? Aquaporin is an example of facilitated diffusion, where it just stays there, but based on the gradient, based on the difference on concentration, it can go in and out, right? So that's facilitated diffusion. An example is water in case of aquaporin. Another example is glucose, right? Those are the two examples with facilitated diffusion. Let's go to the next one. The next one is active, right? Active transport, but this is primary active transport. So notice that during diffusion, we don't require energy. We passively going through, whether going through the membrane in terms of simple diffusion or going through a transporter or a channel in terms of facilitated diffusion, but there is no energy. But whenever you get active transport, we need energy. Now, why do we need energy? It is because we're going against the gradient. If you're going against the gradient, you have to push. Something's gotta push. And we use energy, we use ATP. So this is a primary active transporter. All right. So an example of active transport is this sodium potassium ATPase, right? Sodium potassium ATPase, look at how it works. It's going to actively transport sodium and potassium just like that, right? It needs ATP. ATP comes in and moves this, right? ATP comes in and changes the conformation of the protein, and through that, we're going to have things go in and or out, all right? Active transport. So example is sodium potassium ATPase is a big one. Calcium ATPase is another big one as well, all right? So on top of primary transport, we get secondary transport. Primary active transport, then secondary active transport. How does this work? It's active, so it's also working against the electrochemical gradient, right? But it is driven by something else. What do I mean by that? The movement of an ion, right, or the movement of something is going to drive that active transport. So it's secondary active transport. So you have two things happening. Let me give you an example. An example is sodium hydrogen transporter. By the way, these can be two types. So you see how these two things are going in at the same time? They can be going in at the same time, or one can be going in and the other one going out at the same time. That's the difference between an antiporter and a symporter. So this is an example of an antiporter. Is going to get in, so you have, so look, these one goes in, the other one goes out. So this is sodium and hydrogen antiporter. One goes in, the other one goes out. This is secondary active transport, right? And of course, this is also using energy. And then another one is sodium glucose symporter. The symporter is the one where they are both going in the same direction, right? This is a sodium glucose symporter. All right, so they both go at the same, in the same direction, right? It's a symporter. So you have an antiporter and a symporter, right? So an example, another example in your book is ACARB, which is a secondary active transport system, and H plus drug antiporter is another example. So we gotta know symporters and antiporters and the difference between the two, all right? So that is secondary active transport. You have two types of this, secondary active transport, All right. So the next one is an ion channel, right? Ion channel. Ion channels are also important, right? So as let's look at if you look at a kind of something that's similar is facilitated diffusion. Facilitated diffusion is a diffusion that's facilitated, but it drives non-polar molecules, right? But ions have to go through. And sometimes ions go through active transport against the electrochemical gradient. But when you're going down the electrochemical gradient, then we use ion channels. Ion channels. How do ion channels work? Well, you've heard of sodium and potassium ion channels, and ion channels can be gated. They are usually gated, so they open and close, which is really cool. But this is a sodium potassium ion channel. This is a good example of this. So you have sodium and potassium ion channel, right? The gate is closed. This is what happens in the brain cells, by the way. If you've learned about polarization, depolarization of the brain cell, this is what happens to change the concentration of sodium and potassium along your brain cell, and that's literally how we process information. Because this is how we move electricity along your neurons, right? So whenever you have depolarization, it's going to transmit a signal to open the gate. And guess what happens? It goes through, right? The ions go through, and then whenever that happens, potassium channel opens, so the potassium also opens as well, right? But then whenever you start repolarizing, then you're going to start closing the gate, right? Repolarization is whenever you close the gate, the sodium gate, the potassium channel is open. This is repolarization, right? So this is what happens to propagate the electricity along your nerve cell. But we're focusing on the channels, the ion channels. They can be gated or non-gated, but this is a way to move ions along or down the concentration gradient. We use ion channels, all right? So those are examples, ion-gated sodium channel and potassium channel. Another example is GABA. A GABA channel is also another ion channel. All right, so let's look at another example. Another type of transport is an ionophore. Ionophore transport. This is an interesting one. It's less common, but it happens where to transport something across the membrane, well, if an ion doesn't have a specific transporter, how does it go to the membrane? Sometimes it can hitch a ride on something called an ionophore. And an ionophore is just something that binds to an ion. So an ionophore binds to an ion, goes through the membrane because it can, and then when it gets inside, it releases the ion. And this is because, by the way, the membrane is non-polar. The membrane is not going to be nice to the ion, so it can't just diffuse like non-polar molecules can. So this is another example, another illustration for it, where something binds to the ion and then brings it through. So this is an ionophore versus a channel. So B is an ion channel, A is an ionophore, right? So an example is chloroquine. Chloroquine binds to the zinc ion and then brings it inside the cell, whereas the zinc ion by itself couldn't get through the cell. So chloroquine is an ionophore of zinc, and then it just brings it into the cell. All right. So these are the six types of ion transport. But by the way, remember six types, but secondary active transport has two types. So that's seven types, but I have one more. I have one more to show you. What's one more? Is the vesicles. Vesicles, right? Vesicles are another type of transporting things inside and out of the membrane, right? So you've heard of endocytosis and exocytosis, right? So some things, in order to get them out of the membrane, we make vesicles out of them, right? We make vesicles out of them, and then these vesicles are going to fuse with the membrane and then get out. Or endocytosis is the same thing. You bring something in here, instead of just going through, it goes at the border of the cell membrane. We make a vesicle, and then it goes inside and it forms a vesicle like that, right? So this is another way the cell uses to transport things in and out of the cell. All right, so we just looked at eight eight different uh, eight different transport modes. Okay, so make sure that you know about them. Make sure that you know how to describe them. Make sure that you know at least one example of for each. Make sure that you know how they work, and that is enough for today. And I'll see you in the next one. Bye bye.